Hello, everyone. I'm Qinyu from Brigham Young University, and I work with Dr. Douglas Tree. So this is a recorded version of my APS March meeting presentation this year in 2021. So the topic of my presentation is using reactive dissipative particle dynamics to understand local shape manipulation of polymer vesicles. So the idea of our research is inspired by the concept of morphogenesis. It's a biological process that describes the behavior of organisms taking forms and changing shapes at different length scales and levels. So this process involves various of regulatory factors, both genomic and extragenomic, and it requires numerous of information exchange roads, making it hard to define a definite physical mechanism. So it's kind of natural to think of simplifying and mimicking this process in a synthetic system. So if we can achieve morphogenetic behavior in a synthetic system, we could potentially understand more extensively about the morphogenesis in biological systems and also apply it to different research areas. For example, we can synthesize artificial tissues and make protocells that can divide and replicate autonomously for self-repair, or create certain patterns and use it in mini robot manipulation. So among all the potential candidates, responsive block copolymer vesicle is of particular interest because it has a similar basic structure to the biological cell membranes, and they're relatively easy to prepare in labs. There are several types of external stimuli. The most commonly used ones include pH, temperature, and light, etc. So if we can keep the stimuli in a localized position, we might be able to observe localized deformation or spatial migration of the vesicles. So in today's talk, we will focus on the study of localized shape change. So we propose to study the local deformation using a minimal protocell model, which consists of a block copolymer vesicle and incorporated enzymes or external stimuli to give rise to certain chemical reaction. And eventually, the reactions would result in certain forms of local shape change. So in order to simulate such a system, we would need a coupled reaction diffusion model that can simulate the motion of particles in the polymer solution, as well as the dynamic changes during the reactions. So we first picked dissipative particle dynamics as our diffusion model. DPD is a coarse-grained model with pairwise interactions between particles, and it also includes the hydrodynamic interactions that is important in vesicle formation. So the basic DPD model includes three different interactions. The conservative force is a soft repulsive interaction that allows overlapping of particles. And AIJ is the interaction parameter that controls the miscibility of different species. The dissipative force accounts for the drag of fluids and slows down the particle motion. And the random force is responsible for the thermal fluctuation of the system. So these two forces work together as the thermostat of the system. So these three pairwise interactions vanishes when the distance between the two particles are beyond the cutoff radius. And for polymer chains, we added a harmonic bonded interaction between neighboring particles within the chain. So accordingly, we picked a particle-based stochastic model called split Reactive Brownian Dynamics to simulate the reaction kinetics. The SRBD model stems from the collision theory, which assumes that when two particles come into contact, there's a possibility that the collision will trigger a reaction. So the basic algorithm of SRBD is that it divides the simulation space into subvolumes and accounts for reactions that happen within neighboring cells. So for each cell, we calculate a reaction time based on the number of reactive particles in the neighbors. And then we set up an event queue by arranging the cells according to their reaction time. So at each iteration, we either update the reaction queue if reaction happens, or remove the first cell from the event queue if reaction does not happen until the time reaches the next time step, or the event queue is empty. So we developed a Python code based on the models that we just described and compiled the code using an open source compiler called Numba. Together with its compatible CUDA programming feature to perform GPU accelerated computation. So more of the performance details could be found in our recent publications. So now with the code, we explored two possible local shape change mechanisms. First, we observed local solvent swelling driven by membrane-embedded enzyme particles that catalyze the conversion of surrounding solvent particles into another solvent, which is shown in red, that interacts favorably with the solvophobic particles. So the figure shows a time series of this reaction in which we can observe a clear local swelling to the initial, when we compare to the initial vesicle structure, because the converted solvents tend to diffuse in and mix with the solvophobic particles. And the extent of deformation increases with the simulation time due to the rapid production of converted solvent. We then look at the microinjection case where we instantaneously convert a droplet of solvent particles at the beginning of the simulation. So note that this series of simulation is significantly longer than the previous one, since we want to observe a full range of deformation. So in this case, solvents are all joined into the vesicle, and this leads to a similar swelling in the outer layer of the vesicle as in the previous case. But at long times, the converted solvent diffuses more evenly throughout the vesicle membranes, and the vesicle returns to its spherical shape with slightly increased size due to the solvent swelling. This is also quantified by the change in aspect ratio, which is defined as the ratio of spans of two orthogonal directions as a function of simulation time. So the aspect ratio curve has a trend of decreasing and then gradually increasing back to the point before reaction, suggesting that the solvent swelling happens more globally and uniformly with increased simulation time. Then we explored another mechanism which introduces a kind of external stimulus, which is shown in orange, to convert the solvophobic particles into solvophilic ones. So the figure gives snapshots of a typical simulation as a function of time, and the pink particles represent the converted solvophobic particles. As we can see from the figures and videos, this change results in a similar local deformation to the solvent swelling case. But this is due to the fact that the converted particles tend to diffuse outwards and stretch into the surrounding solvents. So in this case, we also performed a fast kinetics simulation where we instantaneously changed the block fraction of about 4% of the total chains in the vesicles and ran a longer simulation to observe the full range of dynamics. So the snapshots on the right show that vesicle remains largely spherical, but there is still an increase in curvature of the outer corona region where the chain block fractions were, the chain block fraction was altered even at t equals 25,000. So this suggests that this mechanism could provide a more long-lasting local shape change compared to the solvent swelling mechanism. We then compared the diffusion time of a non-bonded solvent particle and a bonded polymer chain. So the figure on the left shows the mean square displacement as a function of simulation time. It is obvious to tell that the mean square displacement of a polymer chain within the same period of time is significantly shorter than that of a single particle. And the fitted diffusivity of polymer chain is about 15 times lower than that of the solvent particle. The videos on the right might give us like a more straightforward demonstration of such difference. While the single particle has diffused across the simulation box for a couple of times, the polymer chain is like still wiggling around its like initial position. Therefore, we think that the relatively longer diffusion time of polymer chain result in the more persistent local deformation. So to sum up, we have developed a GPU-accelerated reactive DPD code that could simulate stochastic reactions in a polymer system. So with this code, we investigated the morphology change of polymer vesicles due to either solvent swelling or changing the polymer properties. Our results suggest that changing polymer properties would render a more persistent local shape change, which we think is due to the longer diffusion timescale of polymer chains relatively to the non-bonded solvent particles. So in the future, we would like to study more complex morphological changes in the vesicle systems, probably with reactions that happens inside the vesicle lumen. Directed motion is another potential topic to explore with our code. So with that, I would like to thank the funding support from BYU College of Engineering and Simmons Research Endowment, and also the technical support and computational resource from BYU Office of Research Computing. And I would also like to thank all of my group members for their help and support. So thanks everyone for your attention, and please feel free to contact us if you have any questions about this presentation or about our paper. Thank you so much.